Hi, good afternoon, everyone. Thank you very much for inviting me to give a talk on our recent work to characterize human exposome using single and multi-omics approach. We also want to identify mechanisms underlying the environmental signatures of diseases. My name is Donghai Liang. I'm Associate Professor of Environmental Health and Epidemiology from Emory University. I'm currently directing the Environmental Metabolomcs and Exposomics research group at Emory Emerge. And in the past decade, we have been applying high throughput omics technology and exposomics profiling, trying to explore the mechanisms underlying the toxicity of different environmental pollutants shown here on several vulnerable populations, including maternal and child health, vulnerable population who live with chronic diseases such as Alzheimer's diseases and cancer. The motivation for us to conduct this research realizing the serious public health threat caused by environmental pollutants. In traditional environmental health research, we often rely on investigating the overall association linking a specific exposure directly to a health outcome. But these correlations alone do not tell us how and why an exposure will lead to diseases. The question mark in the middle of the diagram shown here represents a crucial gap, namely the underlying mechanism. So to truly understand and intervene, we need to move beyond association and use system-based and omics-driven approaches that can uncover the molecular pathways and mediators bridging exposure to health effects. And these challenges are even more difficult to solve in the field of exposome research. As you all know, the exposome represents the totality of environmental exposure we encounter across our life course. So in the paper published by Ru and Gary back in 2020, you will see that everything from the air we breathe in, the water we drink, and the food we ate to the social and occupational environment all compensate the lifetime exposure. So this including complex mixture of physical, chemical, and psychological stressors. And this exposure usually they don't act alone. They interact with our biology in complex and dynamic ways. They shape our human genome, epigenome, transcriptome, proteome, metabolome, lipidome, and microbiome closely. The big challenge we face nowadays in the exposome research is how to link this vast diversity of exposure to measurable molecular changes and then link them to the health outcome that we observe in epidemiological studies. So to tackle these challenges, one of the more widely used approach is the so-called meet in the middle framework, which is designed to go beyond simple association by identifying molecular signatures and pathways that are shared between exposure and health outcome. And in other words, instead of only asking whether or not exposure will lead to a diseases, we oftentimes look for biomolecules such as metabolites, proteins, DNA methylation marks that appear to bridge both the exposure and the outcome. And these shared biomarkers might give us clues about the biological intermediates that might lie in on the causal pathways. And this framework not only strengthens our evidence for causality, but also provides some mechanistic insight in how exposome factors influence health outcomes. So now I would like to transition in some of the case studies from our own group on maternal and child health to show you how we do this. So as we all know, pregnant mothers and newborn babies are among the most vulnerable group to various ubiquitous environmental pollution ranging from air pollutions to persistent organic pollutants. And this exposure has been linked to numerous adverse health effects. Unfortunately, this health burden is particularly affecting people of color, specifically Black and African Americans. They are constantly exposed to higher environmental levels of pollutants, as well as experiencing highest risk of adverse birth outcomes such as small for gestational age and preterm birth. Despite these evidences, the underlying mechanisms are largely unknown due to the lack of sensitive biomarkers, which greatly limited the development of effective intervention to prevent these populations. So thanks to the development of high throughput sequencing and omics technologies, many of this approach shown here have emerged as promising platform that can potentially tackle these challenges. And they have been widely used in exposome research nowadays as an important biomonitoring tool. So for my talk today, I will start with the application of high-resolution metabolomics. So metabolomics is the large-scale global detection and characterization of small molecules called metabolites. As you can see here, compared to other omics layer, the metabolome is really the final downstream product of the gene transcription and gene environment interactions, and therefore the changes in the metabolome are amplified relative to the changes in the genome, epigenome, transcriptome, and proteome. And as the downstream product, the human metabolome is also closest to the phenotype of the biological system, really providing us the most direct functional readout of all the cellular activity and physiological status. So 10 years ago, I started working with Dr. Dean Jones, the pioneer in this field of metabolomics and exposomics, and his team at the clinical biomarker laboratory at Emory has developed this really efficient platform that's capable of detecting and characterizing tens of thousands of these metabolic features from each of the single biosamples. And here shows you a brief overview of how we analyze this metabolomics data using our in-house developed feature extraction methods, followed by metabolome-wide association study, and then coupled with pathway enrichment analysis and chemical annotation, trying to identify all these metabolite and pathway that are associated with environmental exposure and trying to understand their role in causing the downstream health effects. Using this powerful platform, our team has successfully assessed internal exposome and mechanisms underlying toxicity of different environmental chemicals on various health outcomes. I will first start to highlight some of our work where we use meet-in-the-middle approach to investigate association between maternal and newborn exposome, trying to look at the molecular signatures and pathway that are associated with adverse birth outcomes in the Atlanta African-American maternal and child cohort. And thanks to the funding support from the ECHO and NIEHS specifically with this multiple findings. So let me tell you a little bit more about our study population. So this is an ongoing prospective birth cohort with more than 700 pregnant African-American mothers recruited. And as you can see here, this is a socioeconomically diverse and highly phenotyped birth cohort with 19% of the mother delivering small for gestational age baby, with about 13% of the infants born preterm and another 28% born early term. Within this cohort, thanks to the funding support, we are able to comprehensively characterize the maternal and early life exposome using different air pollution modeling coupled with targeted environmental chemicals to assess PFAS, phthalates, smoking, marijuana, different emerging environmental pollutants such as phenols, PAH, PBD, insecticides. Then teaming up with Dr. Dean Jones, Dr. Doug Walkers, and Dr. Alicia Smith, we are able to complete multi-omics data profiling on different longitudinal serum samples collected during early and late pregnancy, as well as with the newborn samples and the early life samples, where we can look at maternal epigenomics, metabolomics, lipidomics, microbiome, coupled with newborn and early life metabolomics. And then we have also comprehensively characterized a range of different clinical data collected and dietary data and behavior risk factor data collected from questionnaires. So based on this cohort, my team has led several major exposomics and multi-omics study. And the first one I want to highlight here is the PFAS exposure and child health study, the PEACH study. Here we focus on PFAS. As many of you know, PFAS is a group of widely used, long-lasting chemicals, and they break down very slowly over time in the environment. So we call them the forever chemicals. So in the past decade, many environmental and molecular epidemiological studies have demonstrated that exposure to PFAS can lead to adverse health effects, including negative pregnancy and birth outcomes. So in this PEACH study, we hypothesized that elevated exposure to PFAS during pregnancy will result in perturbations in specific metabolic pathways, specifically those that are linked to oxidative stress and inflammation, which would then increase risk for adverse birth outcome, including preterm birth and small for gestational age. So 4 years ago, one of our PhD graduate, Dr. C.J. Chang, had published one of the very first meet-in-the-middle approach study trying to understanding the mechanisms linking PFAS exposure and fetal growth in our cohort. To study the underlying mechanisms, we focus on PFAS exposure data and the high-resolution metabolomics data that are collected during the early pregnancy among about 313 participants within our cohort. Then we follow this meet-in-the-middle framework, where basically we conducted 2 independent metabolome-wide association studies, with one focusing on the PFAS levels detected in the serum samples, and the other focused on the adverse birth outcomes. And here we focus on the birth weight as a continuous variable, as well as the odds of whether or not these pregnant mothers will deliver a baby that is small for gestational age. And in this meet-in-the-middle framework, really our task is to identify pathway that are shared between both the exposure and the outcome, and also trying to identify the metabolites that are significantly associated with both the exposure and the health outcome later. So here we observe significant association of maternal serum PFAS concentration with increased odds of delivering baby that are having restricted fetal growth. Where, as you can see here, we are seeing that increased levels of PFOA and PFNA, 2 of the common species of PFAS levels, are significantly associated with lower birth weight. And consistently, we're also seeing a significant positive association between PFAS levels and increased risk of delivering baby that are small for gestational age. Now the question becomes whether or not we can use metabolomics to explore the underlying mechanisms. So here, using pathway enrichment analysis, we successfully identify more than 20 metabolic pathways shown here that are associated with both exposure to PFAS and risk of restrictive fetal growth. Specifically, we found out that this perturbation of this maternal metabolome seems to center around amino acids metabolism of tryptophan, arginine, histidine, lipid and fatty acid perturbation in bile acid, as well as androgenic hormone disruption. Then, compared to the chemical standard provided by Dr. Ding Zheng's lab, we are able to confirm the identity of 10 overlapping metabolites that are confirmed in level 1 evidence shown here. So in particular, I want to draw your attention to uric acids, which is one of our top metabolites. It's a robust intermediate metabolic markers where we found out that during the early pregnancy period, with increased level of PFOA and PFNA, we are seeing that the intensity of uric acid also increases. And with the increases in the uric acid intensities, simultaneously, we are saying that it's associated with a significant decrease in the birth weight of the baby that are delivered by these mothers. And marginally, we also seeing a significant increase of the odds of delivering a baby that is small for gestational age. And not only did we find out uric acid, we also seeing several other potential metabolic intermediates that associate both exposure and outcome. So this includes some other amino acids such as glycine, taurine, and a couple of unsaturated fatty acids, bile acid, and androgenic hormone, which are all closely related to oxidative stress, inflammation, and placenta transport function. And the perturbation in this metabolite can then in turn affect fetal growth. As much as we are very excited about these findings, we know that this is for hypothesis generation. So to validate these findings, Dr. Caitlin Tybalt, a recent PhD graduate from our group, led a follow-up metabolome-wide association study to see whether or not the metabolic patterns that we observe in the mothers can be replicated when we try to focus on the newborn dry blood spot metabolome. And this work was previously published in Nature Communication, and also we want to thank and acknowledge the support from the NIEHS here Lab Hub, because we conducted the newborn metabolomics analysis in Dr. Susan Summers' lab at UNC. So working with the UNC here Lab Hub, we successfully obtained more than 7,000 metabolic signatures in the newborn dry blood spot metabolome. After the metabolome-wide association study, we found out that there are more than 600 overlapping metabolic features that are associated with both prenatal exposure to PFAS and increased adverse health outcomes after adjusting for covariates and multiple testing corrections. So compared to the chemical standard, we are able to confirm a total of 56 overlapping metabolites with level 1 evidence, which can further be grouped into 7 major classes shown here with amino acids, bioacids, and then arachidonic acids consistently showing the strongest association. And more excitingly, 8 out of the 10 metabolites that we are able to confirm in the mothers, again here, are replicated in the newborn metabolome with the same direction of the association and a similar magnitude of the association with both exposure to PFAS and reduced fetal growth. We are also able to identify 14 unique overlapping pathways, and many of these highlighted in red are also the same one that we previously observed in the maternal metabolome. And together, we propose this potential molecular network underlying the toxicity of prenatal PFAS exposure on reduced fetal growth, which center around tissue neogenesis, neuroendocrine function, and redox homeostasis. So building on these exciting findings, we are now conducting additional analysis to systematically unravel the mechanisms linking PFAS to reduce fetal growth and preterm birth using advanced mixture analysis, high-dimensional mediation analysis, and multi-omics integrations. So for instance, 3 years ago, we teamed up with Dr. Stephanie Ike, also from Emory University, and here we conducted one of the very first environmental mixture metabolomics investigation where we use quantile computation to assess the joint effects of PFAS mixtures on maternal metabolome. So unlike most of the existing environmental metabolomic study, instead of looking at each of the PFAS congeners individually, we use QG-COM and for the very first time to look at the joint effects of this simultaneous exposure to 4 different PFAS. And what we found out is that using the mixture analysis, we are able to pick up a stronger and more metabolomics perturbations in the human metabolome during the early pregnancies, which indicated that when we conducted this exposome omics analysis, we should really think about not only just look at single or individual contribution from each of the exposomics risk factor, but also to consider the simultaneous exposure using mixture analysis. And in order to have a more comprehensive view on the molecular network connecting PFAS and adverse birth outcomes, we have also moved beyond metabolomics by investigating other changes in other omics layers. So in addition to metabolomics, we try to investigate the maternal epigenomics by characterizing the whole DNA methylation patterns. So this work is led by my PhD candidate, Yoran Tan, and focusing on the DNA methylation data that are extracted as part of my current R01. Yoran actually analyzed over 800,000 CpG sites using the EPIC-V1 array, and she found out many of the significant associations between PFAS and DNA methylation patterns in the CpG sites that are mapped to genes that are involved in cytoskeletal organization, cell growth pathways, cytokine regulation, cell development, and lipid metabolism. So this result suggests that prenatal exposure to PFAS might also trigger epigenetic changes in the genes that are critical to fetal growth. And using QG-COM again here, when we look at all this PFAS mixture, again, we are finding actually many more CpG sites that are linked to the PFAS exposure, suggests that these exposure mixtures might impact genes that are critical for fetal growth, fetal metabolism, energy metabolism pathway, and cellular functions. And consistent with the epigenome-wide association study modeling, the gene ontology analysis also reveal enriched overlapping pathways that are tied to fetal growth and placenta function, including the top pathway being the IGF binding, the CM signaling, and the steroid hormone metabolism, which were previously observed in the Metabolomics Association study. We also observed several infant sex effect modification, which is quite interesting, where we find out that for PFHS methylation in the TNF RSF1B increased only in the boys, in the mothers who carry a male infant, not the female infant. And we are also seeing a similar sex effect modification in the LRP, which is linked to the lipid metabolism and the TGF beta signaling. Because we are seeing so many lipids getting picked up in both the epigenome Y association study and the metabolomic Y association study, we decided to also look at the lipidomics layer. So this recent lipidomic study led by our PhD graduate, Dr. Caitlin Tybalt, identified numerous lipids in strong association with both PFAS exposure and increased risk for small for gestational age. And again, we are seeing that when we consider all the PFAS mixture together using the QG-COM, we are seeing like a largest number of lipid changes, which are also mostly negative association, which is consistent with the findings that we observe in the metabolomics and the epigenomics layers. And specifically, we are able to identify and confirm 11 lipid intermediates shown here, which links with both PFAS exposure and increased risk of small for gestational age. Most of these lipids were phosphatidylcholine or saturated glycerates, and several of them contain bioactive fatty acids, and they're in strong association with PFNA, PFAS, and PFOA. This figure shows you that there are 12 overlapping lipid pathways, with the top and key perturbation found in glycophospholipid metabolism, insulin resistance, and long-term depressions association also in pattern. And finally, combine all this data together, we find out that these enriched lipids may appear to be highly correlated and interconnected with each other, which suggests that there's a close role of lipidomics perturbation in also shading the inflammation and oxidative stress that will eventually lead to increased risk of adverse birth outcomes. And beyond PFAS, we are successfully also to use this in-the-middle approach to investigating other emerging environmental chemicals and look at their effects on maternal and child health. So this includes our study on tobacco smoke, other emerging pollutants such as PBB, PCB, bisphenols, pesticide, and heat exposure and air pollution. So based on this case study, we can tell that the meet-in-the-middle approach is really valuable for exposome and omics research because it helps identify shared biomarkers linking environmental exposure to health outcome and provide a biologically plausible mechanistic insights. It's especially useful for hypothesis generation. However, meet-in-the-middle approach also has its own limitation because it cannot prove causations. So because most of this study are cross-sectional study, so we need to definitely replicate them into independent studies. So to complement this meet-in-the-middle approach, more recently, we have begun to use another emerging method called the high-dimensional mediation analysis, which really takes the meet-in-the-middle concept a step further by moving beyond individual mediators. So instead of testing one molecule at a time, the high-dimensional mediation analysis actually evaluates the entire layers of different omics data together. So whether there's thousands of metabolites, proteins, or DNA methylation. So here, the high-dimensional mediation analysis will be able to really quantify the relative contribution of each of these mediators and see how they together will jointly mediate the association between exposure and outcome. And so for the last part of my talk, I'm really going to showcase one of the study that we recently do as part of my ongoing R1 study that focus on air pollutions. So as we all know, both short-term and long-term exposure to ambient and traffic-related air pollutants, including 5 particular matter PM2.5, have been associated with numerous health effects shown here. So this includes a reduced fetal growth and low birth weight. And unfortunately, this public health burden is not shared really evenly across the population, with African American and Black American consistently experience the highest risk of having a preterm birth baby compared to other populations. And then due to the lack of sensitive biomarker, the underlying mechanisms still remain largely unknown. So one of the objectives of this particular R1 is really to understanding the biological mechanisms, what's going on within the maternal epigenome, metabolome, and microbiome, trying to understanding the effects of ambient and traffic-related air pollution on adverse birth outcomes. And based on our previous experiences and study, we have already published a lot of articles using metabolomics in assessing internal exposure to air pollution. We are going to use the same workflow, but this time coupled with high-dimensional mediation analysis, trying to understanding the underlying mechanisms in our particular cohort. So here shows you some of the basic demographic characteristic of the first 330 mothers that we are able to investigate in this particular first study. So here, as you can see in the demographic table, you can see that participants who deliver a baby that are preterm or early term tend to have less education compared to those who deliver a healthy full-term baby. We also find out that the participants who deliver a preterm birth baby are also among the mothers that have the lowest early pregnancy BMI and highest proportion of using maternal tobacco and marijuana use. As for the exposure assessment, so for this very first study that we published a year ago, we mainly focused on 5 particular matter PM2.5, and they were estimated using established model developed by our collaborator at Georgia Tech, which have a spatial resolution at 250 meters. So here we consider 4 different exposure time windows ranging from 1 year prior to the conception, averaging through the first trimester, and 1 month and 1 week before the blood draw. So to look at both the short-term and the semi-long-term effects of air pollution exposure. And here shows you the correlations and the distribution of PM2.5 across our study area in the participants. So as expected, as you can see, for many participants who live very close to the highway, they are exposed to a highest level of PM2.5 compared to other participants. And now the question becomes whether or not we can use high-resolution metabolomics to really see the exposure contrast within the internal metabolome. So we use this established method developed by Dr. Dean Jones, and then within their lab, we are able to detect more than 20,000 metabolomic signatures. And then Dr. Zhenjiang Li, who graduated from our group 2 years ago, for the first time adopted a parallel strategy, not only using the meet-in-the-middle approach, but this time he also applied high-dimensional mediation analysis simultaneously, identify and validated mediating metabolomic signatures that are linking the prenatal exposure to PM2.5 and increased risk of preterm birth. So for the meet-in-the-middle approach, it's really straightforward. We start with an NEXUS on the PM2.5 concentration across 4 different exposure time windows, and then we also conducted an independent NEXUS model on the whether or not the mother is deliver a preterm birth baby compared to the full-term baby. So in the meet-in-the-middle approach, we here you can see that we identify several metabolic pathways that are associated with both PM2.5 exposure and increased risk of early term and preterm birth with phenylalanine, tyrosine, tryptophan biosynthesis, as well as protein digestion and absorption being the overlapping pathway. And then in the metabolite level, we are identifying 10 metabolites shown here that are associated with both air pollution exposure and increased risk of preterm birth. I want to draw your attention to hydroxylysine here, because hydroxylysine is not only associated with increased risk of preterm birth, but also associated with all these different PM2.5 concentration across different exposure time window. Besides the meet-in-the-middle approach, as I mentioned before, we are also starting using high-dimensional mediation analysis. Due to the time limit, I want to go into the details here, but we are using an R-package HEMA, which is an established tool to really quantify the mediation effects. And what is exciting about the HEMA analysis is that we are able to really confirm and replicate most of the metabolites that are found in the middle approach and shown here. Here, I want to draw your attention specifically to corticosterone, which really mediates a positive association between short-term exposure to PM2.5 and increased risk of preterm birth and early term birth. We also identify lysopol 23, which also mediate a positive association between 1 month exposure prior to blood draw and increased risk of preterm birth. And again, consistent with the in-the-middle approach, we are finding that like thyroxine, proline, leucine, again, are the top mediators. And using both methods, we are really able to confirm the identity of like 12 metabolites that are shown here that are connecting PM2.5 exposure and preterm birth. And for some of the ongoing analysis, we recently published a paper on environmental science and technology, really comparing the performances of both the high-dimensional mediation analysis and the in-the-middle approach. And we found out that high-dimensional mediation analysis generally provided more accurate mediation quantification by reducing the high-dimensional features, while both the in-the-middle and the high-dimensional mediation analysis really both can capture partial overlapping but complementary signals. So one of the take-home messages for our current work is that in the exposome research, it might be a great idea to use both of these complementary hypothesis generation approach to detect biological intermediates linking exposure and outcome. And moving beyond just one cross-sectional design, we also recently started looking at like longitudinal changes in the maternal metabolomics. And this is one of the papers that it's going to be published in this coming Monday on Nature Health, where my student Yoran Tan look at both the early and late pregnancy maternal metabolome, and she successfully identified several very sensitive metabolite markers that are indicative with great prediction power of like predicting whether or not the mothers later will deliver a baby of preterm compared to the full-term group. And then Dr. Susan Hoffman, who recently graduated from our group, also tried to combine both the metabolomics and epigenomics data together. Long story short, here she identified several CpG size and the metabolomics features that can really tell whether or not a baby is born preterm or full-term apart using these Multi-Omics findings. And we are now trying to validate this in our ongoing analysis. Last but not least, Dr. Zhenjiang Li also using the Multi-Omics approach, also identified several CpG size and metabolites shown here that are not only associated with increased risk of preterm birth, but also they are associated with elevated level of air pollution. And as part of our ongoing NIEHSR1, we are continue generating more of the omics data, and hopefully we can share more exciting exposomics and multi-omics findings with you all in the near futures. And beyond maternal and child health, we also show that this high-dimensional mediation analysis coupled with in-the-middle approach can be applied to study other chronic diseases ranging from asthma, cardiovascular diseases, cancer, and Alzheimer's diseases. And I just want to use like 2 minutes to quickly go over this really exciting investigation that we started like 2 years ago, which focused on Alzheimer's diseases. And as many of you know, in the US alone, Alzheimer's disease affect more than 7 million people, and then they lead to like cognitive decline and long-term care dependency. And Alzheimer's diseases is not only just influenced by genetic risk factor, but they're also really impacted by a lot of these like environmental risk factors that each person can experience through the life course. And supported by several major UO1 and R1 awards from the National Institute of Aging, we launched the IMPACT ADRD study 2 years ago using 3 different well-characterized cognitive aging cohort within Emory. And we are also one of the UO1 study as part of the consortium teaming up with Dr. Gary Miller, Dr. Ding Jones, and Dr. Aimin Chen from like University of like Pennsylvania and Columbia University, trying to really use like multi-omics and exposomics to understanding like the role of environment, its contribution to Alzheimer's disease projects. And right now, working closely with Dr. Anka Hughes and Thomas Ringo, my MPI in this grant, we have shown that like higher exposure to air pollution really seem to increase the risk of Alzheimer's diseases through perturbation in DNA methylations, protein, and metabolome. And then as part of our ongoing UO1, we are also right now trying to comprehensively characterize individuals' exposure using the external exposure assessment by teaming up with Dr. Ted Russell and Yang Liu using like remote sensing and air pollution modeling to look at the external exposome. And we are also teaming up closely with Dr. Dean Jones and Doug Walker using this high throughput exposomics and metabolomics profiling to identify the exogenous chemical into the human brain tissues, spinal fluid, and blood. And then I recently presented some of our initial findings in the Global Exposome Summit, when we have identified several exogenous exposomics factors and endogenous metabolite in the brain tissues in strong association with Alzheimer's disease pathology markers. And we are now trying to replicate these findings in some other biometrics like blood and spinal fluid, and we will keep you posted on our ongoing multi-omics investigations. And finally, we also plan to use a really similar strategy to understanding the exposome's role on lung cancer, and this is one of the studies that I recently proposed teaming up with Dr. In Wang from American Cancer Society. So with that, I want to conclude my talk by really thank my great group at Emory, as well as our wonderful collaborators within Emory and outside Emory. I want to thank all the funding agency in making our study possible, and every single study participant within this cohort for their contribution to the science. So thank you.